Hi, everyone. Welcome to our STEM Festival Day Part 2. Today, we are here on a conversation on how to make a vaccine. My name is Terry Taylor. I'm with the Reginald F. Lewis Museum, and I want to thank you for coming out today. This program is being hosted by the Reginald F. Lewis Museum as well as STEMcx. This is one of our first partnership activities, and hopefully many more to come. Also, this is a program that is being partnered with the Maryland STEM Festival. They are having activities throughout the month as well. Various STEM activities programs are taking place, and I think STEMcx is having another one tomorrow on October 4th. Sandra may tell you a little bit about that in a couple of seconds. At this time, I want to say we're just excited to have a program today about science and making vaccines. And hopefully, we're hoping that this will inspire you to explore maybe a career or um, hobby in science, and hopefully to maybe help make your community a better place. Um, Also, what I would like to do at this time is I'm going to give it over to Miss Sandra Adams today. I'm sorry, to give her general greetings. Take it away, Sandy.
Great, thank you very much and welcome everyone. We are so very excited here to have some rock stars in the vaccine and immunology world here to talk to you. Dr. Kizzmekia Corbett, you probably heard in the news, and she's always out there when they're talking about the COVID vaccine. And so she'll be here to talk to you. Please feel free to put your questions in the chat, or I think Terry may be opening up to let you all ask questions directly to the doctor. We'll also have wonderful rock star fellow Natalia from NIH, and she's going to take us through an activity. So I'm very excited. Like Terry said, we're wearing a couple of hats today. We're STEMcx, and we're also the Maryland Science Festival. So our poster here, definitely check out all the wonderful free activities they have. As Terry said, we have one tomorrow on aerospace. So please sign up if you haven't already done it. So we're going to start off today, since the overall theme is on manufacturing. We're going to start off today to talk a little bit or show a little quick video on design, the manufacturing for vaccines. And as soon as that video is done, we're going to jump right in and talk to Dr. Corbett. So take a listen.
Hi guys, it's so nice to be with you and to see that so many of you are here with us on a Saturday afternoon. I don't remember what I was doing between the grades of 3 and 8 on Saturdays, but I'm sure it was not learning about coronavirus vaccines. So thank you so much for joining me today. I am Kizzmekia Corbett and I work at the National Institutes of Health. Many of you I know are from the Maryland or DC area, and so the National Institutes of Health is actually a part of the federal government. And so we use taxpayer dollars to make vaccines, and overall, the National Institutes of Health uses taxpayer dollars to just make health outcomes for humans so much better. So I am a scientific lead of a team that is developing a coronavirus vaccine that hopefully one day all of you will have the chance to take and will be able to prevent you guys from getting infected with coronavirus. The video that you guys saw was pretty straightforward, but I wanted to talk a little bit about how we do things at the Vaccine Research Center in particular. The Vaccine Research Center is this building right here on the left-hand side, and it is erected under the Clinton administration actually in the 2000s, and only solely for the purpose of making a HIV vaccine. Since that time, though, we've had vaccines go into clinical trial for a number of different illnesses that you guys may be familiar with. But because we're in this pandemic right now, obviously the most familiar to you and the reason why you're on this conference on a Saturday is because of coronaviruses. So the coronavirus team is the team that I lead at the Vaccine Research Center under my boss, who is Dr. Barney Graham. And the very cool thing about our center is that even though it's this one building in Bethesda is a very small building, we have lots of capabilities to make vaccines under this building. And so we can take a vaccine all the way from an idea. In my team, a lot of times those ideas are actually just proteins like you see on this side of the screen, which is a coronavirus protein called the spike. We design those proteins in a very, very, very high level. I don't know if you know what atoms are, but they are very small particles. And we piece those proteins together, and that's how we make vaccines. We also have a pilot plant, and then we also have the ability in a hospital that is on the same campus as us called the Clinical Center. It is the largest hospital in the world that's dedicated to clinical research. We can move our vaccines into the clinic, and then taking cells or blood, etc., from the people who get the vaccines in an experiment, we can test the vaccine and see how well it's working. And after we know that, we can take it into advanced development, which is what's happening right now with our coronavirus vaccine. So I have a small video that I want to show you just so you can understand how coronavirus works. I think it's about 1 minute long.
Coronaviruses invade cells through so-called spike proteins. The spike protein is the major surface protein that binds to a receptor, which acts like a doorway into a human cell. Once the spike protein and the surface of the human cell membrane fuse together, the genome of the virus is able to enter the human cell and begin infection. Understanding the shape of the spike protein in COVID-19 is the key to figuring out how to target the virus and accelerate potential vaccines. Jason McClellan and his team at University of Texas at Austin, in collaboration with the National Institutes of Health, are credited with this important step, which may help in the development of a vaccine against the coronavirus.
So just like that video said, our entire purpose is to understand this protein that is on the surface of a coronavirus. That protein on the coronavirus surface on the left-hand side of your screen is called the spike protein. It is actually a pretty big protein, but on a microscopic level, you can't see it, obviously, with your naked eye. And so we use technology called cryo-electron microscopy to look at that protein in very, 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 very clear detail. And that allows us to be able to see what parts of that protein we would like to target so that that protein won't be able to bind to the host cell inside of the body. So once that protein binds to a host cell inside of the body, that is what allows the virus to essentially spit its genetics inside of the host cell, and the virus can then replicate and cause more viruses to come out of the cell, and that's how people get sick with coronavirus. So obviously, the point of a vaccine is to make sure that we can stop that interaction. We do not want that protein to bind to a human cell. So how do we do that? We trick the body to learn the sequence of that protein so that the body can make some other proteins that are called antibodies. And once those antibodies are made by the body, then the antibodies can bind to the surface of that protein and stop the protein from touching the human cell. So it's almost like creating memory for your immune system. So what's really cool about our particular vaccine is that it is called a messenger RNA vaccine. And I don't know if you guys have gotten to the point where you're learning about DNA and RNA, but messenger RNA is a molecule that tells cells what to do. It messages to cells. Think about it like calling up a cell and saying, hey, cell, make this protein. And so the way that we trick the body to make the spike protein is by making a vaccine that makes the code of the coronavirus spike protein. The very cool thing about the spike protein that we're using is that we've been studying it for so long, not necessarily in the context of this novel coronavirus, but in the context of other coronaviruses that infect humans, like the ones that cause the common cold every year, like HKU1, for example. So we knew so much about that protein that we were able to use messenger RNA once the sequence of the new the new coronavirus came out, and put that messenger RNA in something that's called a lipid nanoparticle. Basically, it's just a circle that covers the RNA so that you can safely deliver it to the body. And then when you get a shot of that RNA, your body cells are being messaged to say, hey, make this spike protein. So you're not getting the virus, you're not getting any other parts of the virus, you're just getting that one protein. And then when your body makes that protein, it starts to learn how to make antibodies. So those antibodies sit around in your bloodstream, they don't do any harm to you. But if you ever come in contact with a coronavirus later in your life, then your coronavirus won't be able to get inside of your cells because your bloodstream has these antibodies that are basically looking out and protecting your cells from being infected by the virus. And so that's how we make a vaccine, particularly for coronavirus. And I will, I'm not sure if I'm taking questions now or if I'm taking questions later, but I'm happy to take them now.
We can take questions now. If you have a question, just use your raise your hand feature in the chat box and we can unmute you. Okay, I see Ashira Evans has her hand up. Let's see.
You hear me now?
Oh, yes.
Okay, so if you get the shot, would it be a yearly thing or every 6 months thing?
That's a very good question. And you know, the reason why people ask that question a lot is because everyone is used to getting their flu shot every single year. Every single season, you have to go to the doctor, actually right about this time, to get your flu shot. So coronaviruses and flu viruses are extremely different viruses. So the reason why you have to get a flu shot every year is because the flu virus has those similar proteins on the surface, like the spike protein, but these proteins are called the hemagglutinin or the HA protein. And the reason why you have to get a flu shot is because HA proteins change with different strains of the flu. So one year there is a flu virus that has one kind of protein on the surface, and then the next year the flu viruses, because they've been circulating around the world all of the year, they come back around and they're completely different. And so now your body doesn't have that memory anymore. The difference here is that the coronavirus that's circulating, or the coronaviruses actually that are circulating, people like to say, oh, they're mutating and they might be different strains, but actually they're so closely related that the spike protein that you'll get from the vaccine, when it becomes available, will hopefully protect you for a very long time. We don't foresee you having to get the vaccine even every year, and hopefully more than that, up to 5 or even more, like a tetanus shot, for example, where it's like a 10-year booster. And so it won't be like how you experienced a flu shot, hopefully. That was a
Great.
great.
Yes. Let's see. Do we have any other hands up? Yes, we have Joan.
Hi, Joan.
Joan, you're unmuted.
Oh, okay. I didn't have a question.
Oh, okay. Well, hi.
Go ahead.
All right, okay. Oh, my question was first because when you showed the video, I couldn't see it.
Okay.
So I didn't know if we were just to listen to it or, actually see it. But I did hear it and I understood what he was saying.
Great. Okay. Anyone else that has a hand up before we move forward to the next thing?
Let me take my hand
I'd like
down.
to ask a question for Dr. Corbett. How do you get involved in this kind of work if you're interested in it?
Well, the first step is to do things like you're doing now, to learn about it and talk to people who are doing this kind of thing. What's really cool about my career is that I started working in a lab when I was only 16 years old. So I'm from North Carolina, and I did research at the University of North Carolina in the summers, in a chemistry laboratory, and I really, 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 really liked it. So as you get older, you should look into labs that may be able to take, for example, high school students. Usually you have to be 16, sometimes 17. But the earlier you can get involved in doing research and understanding research and learning what the life of a scientist looks like on a day-to-day basis, I think it's very important. Not only that, but there are even science experiments and science fairs that children can take part in when they are even in middle school. I mean, I took part in science fairs when I was in elementary school. So researching those science fairs and coming up with a really cool question to ask, it could be a very simple question. I remember asking questions like, how much bacteria is left in your hands after how long you wash them, for example. And you can really design really cool experiments that you can do under parent supervision just at home. And that'll help you to see whether or not you like science, or whether you don't like science. And then the other thing is that to every time you have some level of curiosity about how the world works, the really cool thing about science is that for every fact in your science book or every fact that your science teacher teaches you in school, someone had to discover that. an experiment or several experiments had to be done in order to learn that fact. And so a very cool thing about the internet is that when you learn a fact, like, you know, plants do photosynthesis, you can look online and find out what kind of experiments were done to know that fact for sure. And so that can help you to really kind of engage yourself in the scientific process that are behind all of the facts that you learn from your teacher.
Great. I think we might have a hand up for Michelle Weaver. I want to see if she had something to say or not.
Hi, we just had a question about maybe re-showing the video since we couldn't see it either. That's all.
Oh,
The
no
what?
one?
I apologize. I saw the hand up. Okay. Sorry about that.
Was it that no one could see the video, or did you guys see any of my PowerPoint at all?
Yes, we saw the first sheet of the PowerPoint presentation.
Oh, just the first?
Yes.
Huh, interesting. Well, let me try to find the video via another means, but I agree, you guys should see it.
Okay. Well, would you like to, let me ask you, it's up to you, Dr. Corbett. Did you want to take a couple minutes to see if you could find it, or did you want us to go to the next part and then to come back to you? Either way is fine.
I can, let me see if I can show it now.
Okay.
But yell out if you guys can't see what I'm about to show, okay? Can you guys see that?
Yes.
All right.
Coronaviruses invade cells through so-called spike proteins. The spike protein is the major surface protein that binds to a receptor, which acts like a doorway into a human cell. Once the spike protein and the surface of the human cell membrane fuse together, the genome of the virus is able to enter the human cell and begin infection. Understanding the shape of the spike protein in COVID-19 is the key to figuring out how to target the virus and accelerate potential vaccines. Jason McClellan and his team at University of Texas at Austin, in collaboration with the National Institutes of Health, are credited with this important step, which may help in the development of a vaccine against the coronavirus.
Okay, so that's
Awesome.
it. Sorry that you guys didn't see that before.
Were you able to see it this time, everyone?
Yes, it came out clearly.
Great.
Can I ask Dr. Corbett one more question before we move on?
Sure.
Sure.
Some of the people watching may be a little afraid that when this vaccine comes out to actually take it. So you were talking a little bit about the extensive testing process that can make people feel a little more comfortable taking a vaccine. So what would you say to the people who are a little afraid?
First of all, I would say that it is your right to be afraid of the process, especially because for so long the process has been in a vacuum, and also for so long the process has taken several years to develop a vaccine. So the first thing that I want to remind people is that just because the virus came out in, I guess, the end of December of last year in China, and became a pandemic around March, people, including my team and lots of people all over the world, have been studying coronavirus vaccine development for years. So the patent actually for which the vaccine was designed was filed in 2017 or 16 or 17 because we knew how to design a vaccine based on MERS coronavirus and SARS coronavirus and all of the other coronaviruses that had already we know to be effective to humans. And so it wasn't a necessarily so novel that coronaviruses could infect humans. We actually called them poised for pandemic potential because these viruses can jump from animals into humans. And they have done that obviously before because we've been had an outbreak with MERS and SARS before. And so there's lots of long research that has gone into coronavirus vaccine development. We understood how they work in these types of vaccines work in animals like mice and monkeys. And so, you know, when the sequence of this virus came out on January the 10th of this year, we knew what to do. And so part of the timeline for vaccine development, a large bulk of that is spent on just the research part. And then there's the development. But because we had the research so figured out, it shortened the trajectory to clinical trial. And so you hear Operation Warp Speed a lot, and Operation Warp Speed is birthed out of the idea that you can shorten the timeline in the clinic because you can overlap the different phases of the trial by essentially at risk developing the vaccine towards the idea that it might be licensed, which means billions of dollars are spent, but also, you know, millions of lives will be saved because you'll cut the trajectory of the vaccine development down. And so as we were in phase 1 clinical trial, we got, you know, clear safety signals from that. And then phase 2 was initiated, but before the phase 1 is actually completely over. And then from phase 2, you get the type of signal that you want. And then phase 3 can be initiated when it was initiated at the end of July. And with the safety signal and the type of efficacy signal, which means does the vaccine work or not, we're thinking we'll probably get those types of signal at least by the end of November, but hopefully and probably sooner. We'll know how well the vaccine works before the clinical trial is actually over. These clinical trials last for, they're dedicated, people are dedicated to these clinical trials for years, but you can get the clear signals before they're actually over. And I think that's the thing that's not necessarily translated to the general public before this moment. But what you should know is that when the vaccine goes to market, if it does go to market for general public, that hundreds of thousands of people would have been tested. That actually up to this point, you know, around 40,000 people have already been tested and gotten the vaccine. People that are in our families, people that are my friends, people with my boss's family, etc. And so we believe in the vaccine safety profile and how it works, and would certainly not let anybody get it if we did not feel it was safe. And the layers of which vaccine safety is vetted is not just on the level of us, but on the level of, you know, the FDA, etc. There are tons of people that look at this data and determine its safety. You know, there are lots of misconceptions because it's a vaccine that is with a new technology. So mRNA technology is new to the vaccine world. It's actually not new to the cancer therapeutic world, but because it's new to the vaccine world, people have hesitancy around that. But what I will say is that the vaccine portfolio across the world is extremely broad. And so even if you have hesitancy around the mRNA platform, our patent has been used in multiple platforms, Johnson & Johnson's, Novavax, etc. And so pick your sauce, but get your vaccine, and so that you are protected. And understanding, you know, some people deliver their vaccines differently, however it may be, but knowing that all of them together are being vetted in a very clear and organized process. And I would get it. I actually wish that the ethics department would let me just have it now.
Awesome. Awesome. Well, I think we are ready to go to our next part of our presentation. Dr. Corbett, I want to thank you for that information that you shared with us
Sure.
for our first part. At this time, we are going to have Miss Natalia Christian, our second cool scientist, to come and give her presentation on how to make a vaccine.
Hello, hi guys. My name is Natalia Christian. I am a postbaccalaureate research fellow in the National Institutes of Health. I'm specifically in the National Institute for Allergic and Infectious Disease in the lymphocyte biology section. So my project, well, that basically means that I'm being trained to be a scientist like Dr. Corbett. And I'm also studying to go into medical school. So I am pursuing both my medical degree and my PhD. So I'm here today to talk to you guys more about the research side of vaccine development. My lab specializes in a technique called tissue imaging, and I want to teach you guys about it today. So today we're going to discuss one of many ways to understand three things. Okay, number one, how viruses and other pathogens behave. Number two, how our body responds to these pathogens. Number three, a way to develop treatments and vaccines to keep us and our loved ones safe and healthy. All of these things can be accomplished by using a special research approach called tissue imaging. First, we must review the building blocks of life, though. So a cell is the most basic unit of life. Cells are so tiny that they cannot be seen with the naked eye, as Dr. Corbett was saying about viruses. However, cells are so mighty that they make up all living organisms, including humans. So the human body is made up of 37.2 trillion cells. Cells contain our DNA, create proteins, and carry out the processes that give us the energy we need to survive, among other very important jobs. Despite cells being so tiny, we have developed technology to be able to see them. So we must use microscopes to see these cells. So now that we understand that cells are the basic unit of life, let's build a little bit. So multiple cells organize themselves based on the job they perform, forming structures called tissues. These tissues perform unique functions because they are made of special cells. There are four basic types of tissues. There's epithelial tissue, connective tissue, nervous tissue, and muscle tissue. Each tissue type has its own job. Epithelial tissue is made up of epithelial cells. Epithelial tissue is what protects us by covering our body and our organs. Our skin contains epithelial tissue. Connective tissue is made up of many different kinds of cells, including fibroblasts that produce the protein collagen. Together, these cells serve the function of supporting the other types of tissues and helping them carry out their functions. Our third tissue type is nervous tissues. These are made of nerve cells. Nerves carry messages to other tissues, allowing us to think, to eat, to drink, sleep, feel, and ultimately be alive. The fourth type is muscle tissue, and this is made of three types of muscle cells: skeletal, cardiac, which is your heart, smooth. Muscle tissues serve the function of contracting to allow our bodies to move or to pump blood through the body. So then, now that we've covered tissues then organize themselves into organs, like our skin. Our skin is actually the largest organ of our body, heart, lungs, brain, and others. So these organs organize themselves into organ systems like our digestive system, nervous system, and respiratory system. And finally, these organ systems create organisms like human beings and animals. So when we get sick, it's because one or more of these tissues or organs has been invaded by a pathogen. Pathogens are bacteria, viruses, or other tiny organisms that can cause disease. So we just learned from Dr. Corbett about how we defend against viruses using vaccines that stimulate natural protective mechanisms. More about these in a minute, but first let's talk more about one of many research methods used to gain the knowledge we need to make a good vaccine. So we need to study how the pathogen grows and damages our tissues, And because they are so small, we need to use powerful magnifiers, microscopes, to do this. We also need to understand how the cells of our tissues interact with these organisms and how our immune defenses respond, so we can figure out how to get a protective response without actually being infected or sick. So this is what happens during the research method of tissue imaging. We take organs, we slice them very, very thinly, and then we treat the tissues with stains to make the cells visible and look at these stained tissues under the microscope. Finally, we are able to see if our cells are in a healthy state or in a sick state with pathogens. Once we see our cells in either healthy or in either their healthy state or fighting off pathogens, we take pictures of what we see. So developing images of the tissues allows us a sneak peek into life at the smallest level. Imaging helps us learn more about the pathogens that invade our bodies and make us sick, and how our cells respond to these invasions. Through tissue imaging, we are able to see how viruses and other pathogens behave in our body, study their interactions with our cells and tissues, and develop ways to outsmart them. While performing tissue imaging, we are able to perform experiments to test and understand the following. 1, what healthy tissue and cells look like and how they behave. 2, how pathogens spread in our tissues and make us sick. 3, if a treatment will work to kill the pathogen and restore health to our bodies. And number 4, if a vaccine will work to protect our bodies from illness. So within our body, we have an organ system called the lymphatic system. The components of the lymphatic system fight infections and other diseases. The lymphatic system is made of different organs like our spleen and lymph nodes. During an infection or after vaccination, these lymphoid organs develop special sites called germinal centers, where several cell types work together to produce the antibodies that fight off infection and keep us healthy. Germinal centers contain many kinds of cells, but today we will focus on 3. Number 1, B cells, which is a type of white blood cell called a lymphocyte. Number 2, fibroblasts. And number 3, T cells, which is another type of lymphocyte. So to start a response to a pathogen or a vaccine, a special cell type called a dendritic cell captures the pathogen or the vaccine material. After these cells capture the pathogen or vaccine material, they alert the B and T cells that there is an intruder or vaccine and then trigger an immune response. So instead of waiting for a pathogen to make us sick and then cause our immune system to create protective substances like antibodies, vaccines expose our bodies to harmless parts of pathogens so that the immune system gets a head start and makes the protective response before the pathogen invades. So this keeps our cells from becoming infected and causing disease. So now I will show you a movie that our lab created through a special type of live tissue imaging. In the video, we will see, in 1 second, I'm going to have Dr. Corbett share her screen so we can look at the movie of the immune cells. We can't see anything yet.
Yeah, just give us a couple of seconds. There we
There
go.
it is. Okay, so you can press play. So here in the video, we see dendritic cells in green, and in yellow, we see antigen particles, which would be vaccine particles. And in blue, we see the lymph fluid. So if you can see, and if you can play the video one more time when it stops, please. Yes, so you see the yellow is being fed by the green dendritic cells, and they're being captured from this blue fluid so that it cannot invade and continue to spread throughout our body. So this is live. These are our actual cells, and this is actually what goes on. And the way that we actually got this video was through the work that we do in our lab, and it is called live tissue imaging. Thank you so much, Dr. Corbett. So now I will introduce you all to, now I'll introduce you all to our microscope, because remember that cells are so tiny that you need to be able to magnify them, make them bigger to see them. So now we will show you a video of me showing you our microscope, who we have personified our microscope and named her Bertha. I don't know why, I'm totally new to the lab, but her name is Bertha, and we're going to show you more about how we got that very cool movie and how we get all of this information about cells. Okay, so that is all that I have to show you all today, and I will take any questions if you have any.
There was a question, I'm trying to pull that up during your presentation. Someone mentioned, can babies' immune systems handle so many vaccines?
Oh, well, I think this would also be a good question for Dr. Corbett to chime in on. But just like based off of what I've seen so far, just in like shadowing a neonatologist and several pediatricians, there is a vaccine schedule, and they have taken every precaution to make sure that they don't overwhelm the baby's immune system. But what's actually very fun, it's a fun fact, is that as soon as you're born, you actually receive a vitamin K injection, which is something that helps your blood clot that every child receives as soon as they're born, and that's actually a bacterium that we need in order to survive. And so that's something that everybody receives as soon as we come out of the womb. So we have to be slowly but surely integrated with different types of pathogens, whether they're, definitely the healthy kind, but then eventually we're going to experience those that could kind of be infectious. And so we always, like all of, the healthcare, entity is very mindful, because we do want to make sure that people are able to continue on living. But I'll let Dr. Corbett chime in there.
I think that was actually a very good explanation. And so I want to just say something about Natalia. So Natalia is in this stage of her career that's right after you graduate from undergrad. And so if you have questions about why she chose to take a break before going to med school, or how she got excited about science when she was young, that would be really good. So back to what she was saying about the vaccine schedule. You know, the infant vaccinology is an entire field. The guy that I work for, his name is Dr. Barney Graham, and he's an infectious disease specialist, but he specializes actually in infant vaccines. Actually, his claim to fame is the respiratory syncytial virus vaccine that is in phase, I think 2 or 3 right now. And RSV, if you are a parent on the phone, you probably know that RSV is one of those little illnesses that a lot of kids get sick with. Actually, my nephew who's on the phone got extremely sick with RSV before. And so, you know, kids are going to be exposed to things, and it's best that they get exposed to it in a very controlled way through a vaccine, as opposed to by nature. Because when you get a vaccine, at the very least, you can't be infected with whatever you're getting vaccinated for. Also, the way that the vaccine schedule is set up is so strategic that it allows for your immune system to create this memory, what's called memory, in a very, very strategic way. And so, you know, keeping babies healthy through infant vaccinology is an extremely important thing. The other thing is that for pregnant women, it is extremely important that you stay on your vaccine schedule, because via breast milk, etc., a child can feed and get what's called passive immunity from their mother.
Great.
Babies are far stronger than you think, actually. There's no overwhelming a baby.
We have 2 other questions. One was, what color is a vaccine?
That's a good question.
Good questions. I don't know. Is there a color for a vaccine?
You know, I mean, the liquid is generally clear or like an opaque white. And it's interesting because, you know, those colors that you see when you look at those videos or the proteins that I show, for example, those colors are generated. These viruses, for example, and even the slide that Natalia showed, she made those different things on that slide into those colors by dyeing them. So these viruses don't have color. These vaccines don't have color either. Although maybe that might be a way to get kids to take them. Yeah. Whoever said that, you might just discover something.
Okay. Another question was, what goes in the lymphatic system? They want to know what to write down, where it says what goes in the lymphatic system blank.
Okay, let me check. Lymphatic system. Can that person just read the sentence, including the blank?
I'm not sure.
I think they're looking at the activity sheet.
Oh, is it number 4? It might be number 4. I see something about a lymphatic system. It says, maybe I'll have them to type it. Oh, are they unmuted?
I don't think so, but I can pull it up and I can type it into the chat box.
Okay.
And then go ahead.
I'm saying if they could put what number it was next to, that would be helpful. Then there was another question. Have there been cases of certain vaccines leading to other health issues? Either Natalia or Dr. Corbett.
I think Dr. Corbett.
Yeah.
I'll take this one.
That would probably be Dr. Corbett.
What was the question?
Have there been cases of certain vaccines leading to other health issues?
Yes, there have actually. Back in the day, before we didn't understand as much as we know now. That's one of the reasons why my boss dedicated his career to the respiratory syncytial virus vaccine, because the respiratory syncytial virus vaccine that previously, back, I don't even know, I think it was like the 60s, cause people to get sick because of what's called vaccine-induced illness. So there's an entire field of study around that. It's not because, actually, it's not necessarily because of the vaccine. It's because of the proteins that I said that we study. The reason why we study them is because we want to give the body the best picture of what the protein looks like. And the way that vaccine was made, and actually the old ways that vaccines are made, sometimes destroys those proteins in so much that the body is seeing something that is not necessarily good for it. So a lot of times people will say, well, the new technology, it's so new, but understanding that the new technology is new because the old technology is not always the best.
Great. I have two more questions. The number I found out was number 4 in reference to that question about the lymphatic system. And then while you're looking that up, there's a question for both scientists. Why did you get involved in studying vaccines?
I guess, do you want me to go, Dr. Corbett, or you can go? It doesn't matter.
Go ahead, you should answer it.
Okay. So when I was 7, I knew that I wanted to deliver babies. I wanted to be an obstetrician when I was 7. I don't know why. I just always said I was going to be an obstetrician. So I just, I stayed focused on that. Like in high school, I actually started speaking French. So my French teacher, he knew I wanted to be a doctor, and he was saying that there was an organization called Doctors Without Borders in French-speaking countries, because he wanted me to combine my love for French and my love for medicine. And so then I went to college. I went to Howard University, and I was able to study French while studying. I had a minor in chemistry, and I took all my MCAT requirements. So I took up to biochemistry in college, and I was able to do a lot of shadowing. I went to Senegal, and I was able to shadow a pediatrician there, and I saw several births, and a whole lot of other things. And then when I graduated from college in 2018, I actually taught, it's so funny, I taught high school chemistry. I don't know if some of my old students might be here, but I taught high school chemistry here in DC for a year. And but I never forgot that I wanted to go to medical school. And then my research mentor, because I did do research throughout the years, but it was never anything very consistent. But one of my mentors told me about this program at the NIH called NRO, and it is a research fellowship for post-baccalaureate students in the National Institute of Allergic and Infectious Disease. So essentially, my mentor told me that I could possibly get both my medical degree and my PhD as long as I prepare for it. And I didn't know. That blew my mind, because I was always told that I could only do MD or PhD, and I never wanted to give up medicine, but research was always so intriguing to me. So when they told me I could do both, I saw it as a way for me to help even when I'm not physically here on this earth, because if I'm able to create some type of treatment or figure out or engineer a vaccine, then I don't physically have to be here to help people anymore, so it would allow for me to help for a longer time and on a greater scale. And plus, I really love understanding. I get a rush when I understand things. So being able to study something from the smallest level all the way to the largest level really gets me excited. And the work that I'm doing in the lab is I'm studying the Ebola virus. So what we're doing is we're studying how our immune system interacts with the virus, so then we can create treatments and vaccines for that specific virus. And understanding that these viruses that I'm interested in, they affect usually West Africa, Central Africa, a lot of the African countries and third world countries. So I will be able to ultimately, practice medicine and translate, take what I do in my lab and take it into my patients, and just be able to go in between both worlds in that ways.
It's a wonderful story.
Yep, I didn't know if Dr. Corbett wanted to share anything. And we do have another person who wants to verbally ask their question too, Gabriella. So she'll be up next.
I guess I can just quickly say that the reason why I decided to study vaccines is because vaccinology and vaccines are single-handedly the most life-saving inventions ever from a health perspective. They are essentially the main way, outside of public health measures like washing your hands and drinking clean water, etc., that you can prevent disease. And everyone knows that getting a virus or getting a bacteria, getting a disease is bad for the economy, bad for your health, and treatment on the other side of it actually is not efficient, right? No one wants to get sick only to have to get better through medical care. So preventing diseases have saved so many lives. And so I figured that if I was going to make an impact on the world, I would want to be on the side of prevention. And that's why I got into vaccinology. And here we are.
Awesome. Well, I'm going to unmute Gabriella. She has a question that she wants to bring up or a comment. You're unmuted now. Gabriella?
Oh,
I'll give her one more sec. I'll keep that open.
okay.
Oh, we cannot hear you, Gabriella. If you want to just, I guess your thing is unmuted. If you want to type in your question, we still can ask. Try that one more time while she writes her question.
Can I ask Natalia what kind of doctor you want to become?
Oh, so I told you I always wanted to be an obstetrician. That has could evolve into possible neonatal surgeon, possible, but then now that I'm studying infectious disease, I've learned that I could also be an infectious disease doctor. So every physician tells me you're not gonna know until you do your rotations. So I'm just open-minded. I stay tuned. I'll let you guys know when I know.
Okay, well, Gabrielle is still finishing typing up her question. We do have another one, and we'll close out with Gabriella's. This says, this is from William. How is COVID influencing the economy now?
Greatly. It's one of the reasons why there is such an important task to get people vaccinated, because we can't really return back to normal life without what's called 70% of herd immunity. And so what that means is that in the population, 70% of people are protected they have an immune response that can protect them, that ideally comes from a vaccine. And that amount of herd immunity will be able to translate into some sustainable level of lowering the amount of transmission of the virus. The economy is suffering greatly. I mean, I think they estimate that 1 in 5 small businesses will close by the end of the pandemic in the United States. Obviously, that we are in some of the highest percentages of unemployment that we've seen in the last 5 decades. Yeah, I mean, the stock market, you probably hear a lot about that too. The economy is suffering badly from coronaviruses. But, on an aside, just because coronaviruses are respiratory viruses, everyone should know that actually getting sick from the common cold is actually one of the most devastating things that happens to the economy every single year. The amount of time the amount of work hours that are lost, and the amount of hospital emergency room visits that come from just getting the common cold puts an extreme damper on the economy every single year. So that's actually the reason why I work on respiratory viruses, because even though you think of them from an individual level, you think of them as the sniffles and maybe a little bit of a cough, from a global level, they have an extremely big impact.
Awesome. So we do have the other question from Gabby's moms. And it says, greetings from Chile, South America. And thanks
Oh,
for the
wow.
thanks for the webinar. I'm wondering about, you just never know. I'm wondering about the variety of folks taking part in the trials. I understand that they needed more African Americans. How do you ensure that there are a variety of folks from different ethnic backgrounds?
Yeah, we actually, well, Moderna, in the particular case of the Moderna vaccine, if that's the one she's talking about, Moderna CEO made it a point to make sure that the trial was diverse and inclusive so that it mirrors what we see, at least in the United States of America's population. So our population is about 13.4% African American, and I'm happy to report that we are now at 13% African American enrollment in the clinical trial. Those numbers also are mirrored across the different racial and ethnic backgrounds of people that live in the United States of America. And so, you know, the way that we did that is by community engagement, really reaching out to people, helping them to understand how these trials work, helping them to dissect out the safety of the vaccine that we already, how we already know it based on the other trials that have been conducted in the phases 1 and phase 2. And then helping them to understand that, like, a Black woman invented it. So
Yes.
you're gonna be all right.
Yes.
But yeah, you know, from a scientific perspective, I want people to understand that it's not because, you know, from any level of ego that we want to have this, say this vaccine was tested in this many African Americans, like, look at us. It's actually important because people from very different genetic backgrounds can have different immune responses to different medications. And so if you don't test the medication in populations that mirror the population for which the medication will be deployed, you run the risk of actually not knowing enough about how the vaccine might work in minority populations. And so there is scientific rhyme and reason behind it. And if there's anybody on the trial who participated, you know, thank you.
Great.
Yeah.
Also, it's just mentioned what will happen with children in the vaccines. And then also that comment was Black women and girls rock for sure. I'm also African American from Maryland, so it's great to see Black women in science.
Yeah, children in the vaccine. So, you know, even with all of the companies coming together in Operation Warp Speed, risking billions of dollars to start manufacturing even like as early as April on these vaccines, it's going to be a while before we're able to vaccinate the entire country and for certain the entire world. And so there's going to have to be a deployment of the vaccine in a very strategic way. Obviously, firstly, you want to vaccinate people that are at most high risk. Those are physicians, nurses, you know, elderly, etc., that are at most high risk for the most severe disease. And then pare down the vaccinations towards children later, which is also why, while generally we will be running a trial simultaneously in children, we actually aren't at this moment because we need to get enough data to get it into the elderly first. And so there will be trials run in kids, and after those trials are run in kids and it's deemed safe for kids, kids will be added to the population of which are vaccinated. But luckily, kids are definitely transmitting the virus to adults and etc. But luckily, kids are not getting as sick as they generally get with respiratory viruses in this case.
Great. And this is, oh, okay. Still asking about the answer for number 4, just by the way. I'm not sure, so we're going to try to get that.
Guys, this is
But
Alyssa. I figured out which question. So they're looking for the components of the lymphatic system, blank infections, and
Oh,
other diseases.
okay. So that blank is, hold on one second.
and then I have one last question for Mark, and then we're going to close out.
Fight. It's fight. Sorry. Fight infections and other diseases. And please let me
Great.
know if you have any more questions. I will actually just share the key for that worksheet so that you can review your
That would
answers.
be great. So we will email that out to all the registrants. So if you want to try your hand at completing the worksheet, you'll get the key on Monday to be able to check your answers.
Okay. And then the last question to close us out, what risk are there if someone chooses not to get vaccinated?
Well, then you run the risk of not being protected against the virus. I'm a strong believer that people should make people should have healthcare choices and rights all across the board. I'm a strong believer in choice, and I'm extremely liberal about that. So I don't feel obligated to force anyone to get the vaccine. So it is a choice, but understanding that with that choice, you do run the risk of being infected and potentially becoming extremely ill, and not just a lot of the times getting vaccinated is not necessarily about us as an individual. Mm-hmm. For example, I got my flu vaccine like a week and a half ago. And that's because I feel that it's my debt to society to be protected so that I don't transmit the virus to other people who might not be able to get vaccines for health reasons or because they're old or etc. And so getting vaccinated isn't just about protecting you, but it's about protecting the people that are around you. It's almost one of the most selfless things you can do in regards to your own personal health. And then, hospitals are overwhelmed and the economy is suffering and all of these other things that come with it. And so doing your part to become a part of that 70% of herd immunity is how I look at it.
Great. Well, we are at this time, we've had a wonderful hour, of learning about vaccines and just learning a little bit about the type of work you guys do and in the environment and setting that you guys are in. And we've had some really good questions out there. People are definitely interested, and we've had people around the world to come join us and around this country as well. So that's been exciting as well. I want to thank both of our presenters, Dr. Corbett and Natalia, for taking their time today to spend with us, to share what they do. We really appreciate that. And I also want to thank Sandy Adams, who also was responsible for putting this program together for the museum. When we discussed having our first program, it was sort of kind of like, wow, this would be kind of interesting. This is what's going on in the world right now and it's relevant. And so now we kind of have a little bit better idea about vaccines, and hopefully all of us will be getting one in the future once they are ready and available. So what I would like to do at this time, I put in the chat box also information about STEMcx. They have a program tomorrow and other additional fun activities. So if you want to learn about what they are up to, you can go to their website. I'm going to put that back in again. You can click on that link or go to their Facebook. And I'm going to see if Sandy has any last remarks or anything before we close out. We will send a survey out early next week, as well as that information on the activity that was done.
Thank you, Terry. I just want to say this exceeded every expectation I had. You all were wonderful speakers, engaging, answering all of our questions, of which there are many. We were all a little worried with the COVID going around and trying to figure out what's going on the vaccine front, and you all just really hit it out the park. So thank you so much for responding to our inquiries to be a part of this presentation. Thank you so much for letting us see a little bit of that Black girl magic.
Yeah.
We wish you well in your endeavors as you go forward. And Terry, it was just a wonderful performance. I couldn't be happier.
Awesome.
Thank you guys for having me.
Thank you. Yes.
And I want to thank Alyssa Foley, who's in the background, who's actually orchestrating a lot of the technical things that are going on behind. So thanks, Alyssa. Well, with that, I think we're going to close out. So everyone, bye and have a wonderful rest of the weekend.
Bye-bye.
Bye.